Good evening, everyone, and welcome. My name is Alyssa Ballingy. I'm the Senior Market Development Manager here at InoRx. And on behalf of the InoRx team, we thank you so much for joining us tonight. We're very excited about what we have to share with you today. InoRx Academy is our ongoing educational initiative that we're going to start having and have designed for meaningful conversations around clinical trial execution, evolving regulatory expectations, and the practical challenges faced by, by sites and sponsors. Our goal is to bring together experienced leaders and investigators to share insights that are immediately applicable in the real world. We're so happy to have you tonight. Before we begin, those who might have not heard of us or are newer to InoRx, InoRx is the next generation clinical research organization focused on delivering agile, quality-driven oncology and specialty clinical trial solutions. We work closely with sponsors and investigator and sites to enhance operational efficiency, regulatory compliance, and trial execution across our global programs. And to give you a brief overview, we are doing so much in this space, and we are so excited to share some of the amazing speakers we have with us today. We're truly grateful to be engaged with an audience like you all. And I am going to just go ahead and show you a brief video, a little bit more on InoRx.
Clinical research has long promised hope to patients. Yet for many sponsors, the path from protocol to patient has become slower, more complex, and increasingly disconnected from real care. InRx was not born in a boardroom. It was built from inside healthcare. Backed by the SRM Group, we saw where the system was falling short. India, home to extraordinary clinicians, investigators, and patients, has historically been underrepresented in global clinical research. We believe that needed to change. We built a next-generation CRO to increase sponsor value while expanding access for underserved patients. Led in the United States, with clinical execution embedded within India's healthcare system, delivering white-glove service to sponsors and offering patients access to a higher standard of care than they might otherwise receive. For sponsors, this means faster startup, predictable execution, and regulatory-ready data. For patients, it means access, care, and hope. At InRx, clinical trials aren't transactions. They're commitments. InRx, simplifying trials, accelerating cures. Clinical.
Thank you. It is now with my pleasure that I introduce our moderator for today and also our Chief Medical Officer at InoRx, Dr. Melvin George. Dr. George has served as the head of SRM Center for Clinical Pharmacology and the Center for Clinical Trials and Research in Chennai, and also serves as the Chief Medical Officer here at InoRx. With experience leading more than 65 clinical trials and contributing to over 145 peer-reviewed publications, he brings exceptional depth in clinical research, academic leadership, and operational insight. We are so grateful to have him on our team and here today to present with us and moderate for the session. Dr. George, please take it away.
Thank you, Aliza. At the outset, I wish to thank all of you for taking your time this evening to be a part of this InoRx Academy webinar series. We have a very interesting line of topics that we're going to discuss. We all know as in clinical trials, these are the process by which we actually bring out new molecules into the market. And one of the most important players in this whole ecosystem is the principal investigator. So we have here a stalwart principal investigator who has led several trials in clinical oncology over the last few decades. And we also have someone with a vast industry experience to talk about what the industry expects or what are the global standards for an investigator in a clinical trial. We have with us Dr. Raj Nagarkar. Dr. Alisa, could you move forward?
Yes.
Hi.
Yeah, so Dr.
Oops. Thank you.
No. Yeah. So Dr. Raj Nagarkar is a distinguished surgical oncologist who has over 24 years of experience in oncology. He has performed over 50,000 major oncosurgeries, completed more than 2,000 robotic surgeries in just 6 years, which is itself a global landmark. And most importantly, he's been a principal investigator in over 500 clinical trials and over 380 scientific publications. Under his leadership, the Manvita Cancer Center at Nasik has become a 275-bed state-of-the-art oncology center and one of the first in the region to establish a dedicated robotic surgery department, advanced PT, CT services, and is widely recognized for integrating surgical excellence, innovation, clinical research, and patient-centered care while championing large screening initiatives and cancer programs across India. So we are very happy to have you with us, sir. And we also have Dr. Vinayak Shahavi. Lisa, please turn the slide. So Dr. Vinayak is head of clinical operations who has worked with several pharmaceutical companies and contract research organizations over the last 2 decades. He leads global clinical trial execution and operational excellence across different therapeutic areas. He has over 20 years of experience in clinical development and operations and has held several senior leadership positions across CROs and pharma organizations, managing complex multi-country studies in highly regulated environments. And Dr. Shavi brings deep expertise, particularly in oncology trials, regulatory compliance, patient safety, and quality-driven execution, helping sponsors to develop clinical trial programs that are enabled to bring out effective molecules for our patients. Over to Dr. Raj Nagarkar for the first session, where he'll be sharing his vast experience from doing clinical trials in oncology.
Thank you, Dr. Melvin, and thank you, InoRx, for this opportunity. It's interesting more so because it's talking about the real life scenario and the journey from where I started off. Before entering into the private practice, I have worked at Tata Memorial Hospital, followed by my short stint at the Royal College of Surgeons. It was an era before 2001 where clinical trials more so were investigator initiated. Pharma funded trials definitely did exist, but it was mainly from the medical oncology domain. After moving to Nasir, that was in the year 2001, and started practicing as a freelancing surgical oncologist. Definitely, there are a lot of limitations when you are working in a private practice. And I guess today, the August audience and the participants of this webinar, I'm sure some of them would be such who are actually working in private scenario or even in trust or corporate scenarios where clinical research may not be part of their armamentarium on a daily basis. Alisa, can I have my slides, please? Yeah. So actually, if I have to run through my own track record of this more than 2 decades of oncology practice, being a surgical oncologist, all of us are very well aware that we have moved ahead from the era of, or we call it as the Halstedian era. Halstedian era meaning that where surgery was thought as the mainstay of treatment when it came to solid tumors which was there. And if you are talking about breast cancer, then usually the surgery was total mastectomy along with removal of the pectoralis major muscle, minor muscle, the lymph nodes, and even the internal mammary nodes or such. Because the basic philosophy or the understanding was you have to be radical in your approach when it comes to the tumor tissue is such. But over the decades or over the century, I would say that some amount of understanding about the tumor biology actually played a big role in realizing that it was not about the extent of surgery, but it was optimum surgery which actually mattered. And what brought this change? Definitely, it was clinical trials. And these trials were with a very clear foresight that it has to be, that was an era of evidence based medicine. That does evidence really suggest or help us in doing such extensive surgeries with morbidity and mortality? And towards the end of the 20th century, we moved to an era of organ preservation surgery, not only for breast, but even for head neck cancers, laryngeal cancers, stomach cancers, colon cancers, and whatnot. The guiding principle obviously remained that the best care for a patient is not yesterday's standard, but it is tomorrow's evidence. So I'm talking about an era where we were talking about evidence based medicine. In fact, now we have moved ahead from that era of evidence based medicine. We are now talking about precision medicine, that every patient is unique, every patient's tumor is unique. So you cannot have a basic, like, one size fit all approach for all our patients. Next slide, please. Elisa, can I have the next slide? Yeah. So when I started the center in 2007, it started with a team of 35 people. And today, the entire team is of close to 1,500 people who are working, ensuring that how do we make the lives of our cancer patients better. Over these last 18 years, we have treated more than 300,000 cancer patients, and the number of surgeries actually has crossed almost 81,000, not the 50,000 ones which are there. Uh, we have gained significant length in the 500 plus clinical trials which we have conducted, 2,000 plus robotic assisted procedures which have been done. And while this was there, the most important, I still remember those initial days where I was actually struggling to have one clinical research coordinator as part of the team, because I used to read, and I was the coordinator. I was also injecting patient. I was doing the dosing. I was ensuring that all the documentation has been completed. ICFs are filled, and CR. That time it was mainly paper CRFs. All paper CRFs were being entered. So it was like one man army trying to do everything. And the first clinical research coordinator whom I requested and finally convinced to be part of the research team, it would not be an exaggeration to say that I was actually touching her feet that please be a clinical research coordinator, because she felt, sir, I am okay to do she was from a homeopathy background. She said, I am okay to do an RMO ship, but I don't want to be a coordinator who wants to read again, who wants to study again. But fortunately for me, she got convinced, and the journey which started. Today, we have a research team of more than 30 people, and with the ongoing 60 plus trials which are there. The key insight which was always that we always feel that volumes create excellence, but I feel that it's not about volumes. Had volumes created excellence, then probably all our district hospitals, civil hospitals would have been genuinely centers of excellence. What matters is the structured evidence generation also. Until unless you have documentation, see, my own journey of more than 2.5 decades, let me be honest enough, whatever I learned at Tata Memorial Hospital and the Christie Cancer Center, that was a basic platform on which I have gone on to build up my career, my practice, and I have evolved in my patient centric approaches also. What was right yesterday is obsolete today, and what might be right today definitely will be there. There will be a change as tomorrow more and more evidence gathers to make things better for the patients. Next slide, please. So in fact, if you look at the journey from precision to escalation, there has been a paradigm shift, uh, because we no longer treat where the cancer is. We know that any tumor, any cancer for that matter, is a micro metastatic disease right from day one. And I recollect those days when my teacher used to tell us that if you treat the tumor, you will definitely be wrong. But if you treat the biology of the tumor, you will be seldom wrong. So these biological roadmaps, which involved the molecular identity and often guided with the image, not only by the imaging. And over these years, we know now that what in the next genome sequencing, 2 years back or 3 years back, we felt that NGS was a fancy test which is only for a limited number of patients, only for those patients who have failed multiple lines of therapies. But today, NGS has become part and parcel of almost majority of the patients at the time of diagnosis, and it is actually defining the way treatment should be done. De-escalation. The de-escalation protocols have come in, like for example, the dostarlimab study. With the advent of immunotherapy, we now know that do we really need to do extensive surgeries? Use of neoadjuvant immunotherapies, be it in breast cancer or colorectal cancers. The dostarlimab study actually had shaken the entire oncology and non-oncology globe also, because if you have a MSI high patient giving couple of doses of immunotherapy, that's a dostarlimab, it was found that the chances of complete responder was close to 100%. So if you are able to achieve a complete response, radiological and clinical response on neoadjuvant immunotherapy, why do we need to operate? So de-escalation is definitely the key. It is not that being a surgeon, we will be driven out of business. But fact is, we will be able to do more justice to the patients who need that particular treatment rather than performing unindicated surgeries, and definitely the morbidity can be reduced in a good number of patients. Now, the key lesson which is learned over these decades with the availability of all these biological roadmaps is that stage alone does not define destiny. See, molecular signatures are a must. See, we know that till 1990s, a patient with chronic myeloid leukemia, the treatment was melphalan-based chemotherapy. But moment we had the wonder drug, one pill came and Gleevec actually changed the way in which CML can be treated. CML is like, hardly we call about it as a malignancy. It is a chronic disease and one pill is enough to take care of it. The magic bullet was what hit the oncology community, and actually that is how if you can identify the molecular signatures, we will be able to do more justice. Stage 4 breast cancer patients till the era of trastuzumab and pertuzumab were thought as a doomsday call. How many more months are left or weeks are left was the thought process. But now with these anti-HER2 molecules or availability of immunotherapy molecules, even stage 4 patients definitely have a long survival outcome and a very good quality of life as well. Next slide, please. Now, see, it's like, the journey, I would say it is my own journey. And this journey was, when it started, moment we used to say that why not be part of a clinical research or a clinical trial. The patient or the caregiver used to jump up, or for that matter, even doctor colleagues who are not involved in clinical trials would used to have this narration that why are you treating the patient as a guinea pig? Because they felt these were experiments which were being done on patients, and they are, you are just trying to do treatment which is not standard of care. However, when we really put forth our efforts in convincing the patients that it is not about an experiment, but it is a guided way in which the standard of care can actually be defined, and it is completely protocol driven and monitored. Many times, if you are treating alone as per the standard of care, there is always a possibility you might be biased. But when it comes to clinical research, there is a multidisciplinary oversight. There are monitors, medical monitors who are overseeing the progress of the patient, and you just cannot take any patient into a clinical research as per the whims and fancies of that treating clinician. So actually the standard of care honestly tends to be much better as compared to for those patients who are treated outside a clinical trial. So we have definitely gained a good length. We have covered significant miles to understand and explain to the community that clinical research is not an experiment, but it is actually a science, and this science is to just make sure that patient gets treatment which is better than the available standards. Next slide, please. So now there was always a thought process that as Indians, we have been very good followers. So we were very good followers of the West or the East. So even today, many times we are divided whether to follow the East or whether to follow the West, because if it comes to the upper GI malignancy, esophageal or the stomach malignancies, we feel the East is more aggressive and better, but some areas the West is better. And one good example where we failed being followers is the capecitabine example. In fact, all the clinical research studies which were done on capecitabine, the doses which were defined were on an average 1500 to 1800 milligram per meter square. And when we realized hardly any of our patients were tolerating those doses, majority, almost all of them had grade 2, 3 hand foot syndromes. And we realized that the West, in the West, these doses were easily tolerated by the patient, but the Indian subset, we were not good at these doses. It warranted a dose reduction. It warranted a reduction of dose not only for hand-foot syndrome, but also for the gastrointestinal toxicity. Patients who are becoming very, very morbid because of diarrhea, because of nutritional imbalances, and they were not tolerating those doses. And we realized that probably our Indian body habitus, our Indian DNA is definitely different. So there were studies being conducted based on the DPD variability, the body components, the dietary factors, and we realized that the global dosing assumptions are not right for our Indian patients. And we actually went down on our doses and realized our patient outcomes are still better at reduced doses. So here again, it was India-centric approach for the molecule, and we realized that the global dosage and Indian doses are different. Next slide, please. So there is technology and surgical reliability has shown a major evolution. So it's like, I have been trained as an open surgeon, but moved on to the minimal invasive approach, the laparoscopic approach. And now for the last 6 years, I have been focusing on robotic surgery. Earlier, it felt whether robotic surgery will it be a viable proposition? Will our Indian patients accept it, particularly for tier 2 cities? But I've realized that when it comes to technology, the adaptation is definitely very, very good, because you, with MIS, with the laparoscopy or robotic, the ergonomics, the vision is, and the morbidity of the patient play important roles. You are able to do this, give a similar or better outcomes and with a reduced morbidity. With the robotic platforms and so many now coming, it has become a standard of care, I would say, for a good number of cases. And while we are moving now currently with the robotic platform, we know that telerobotic surgery is also there, and very soon, the STAR robot, there's a smart tissue autonomous robot. The John Hopkins Institute conducted the trial and actually proved that a smart tissue autonomous robot can perform independent surgeries. I had my experience in China in November 2025, where I was sitting in Shenzhen and operating on a animal which was in Beijing, that was almost 2,200 miles away. And the lag of the technology was just 0.24 milliseconds. My hands or my eyes could hardly appreciate the lag, and the surgery went off absolutely seamlessly. So technology is there, and I think adaptation is important. It is not that technology or surgical robots will replace surgeons, But I would definitely say that surgeons using technology and surgical robots are sure to replace surgeons not using it. Next slide, please. Now, the Indian genomic imperative I have to put ahead, as I already mentioned, Indians, we just cannot be followers, we cannot be consumers of Western data. Our drug responses and mutation patterns definitely differ. So we must design trials for Indian genes and not just validate Western conclusions. And the global impact which you are definitely expecting, high volume of Indian data must shape global guidelines. In fact, friends, most of the clinical trials which we are part of, we have always remained as the highest enrollers. It's not only about enrolling highest number of subjects, but churning out the best quality of data. The highest quality data is there, and where these things can be achieved in the shortest timelines, wherein the outcomes can be defined, and the clinical trial, even the sponsor and the CROs can achieve the desired outcomes in shortest possible time span, so which can be brought for the patient benefit in real world scenario. Next slide, please. In while doing various clinical trials as a center, we have been involved in a good number of investigator initiated trials, because we have our own postgraduate training programs, super specialty programs. So among the few of the studies which we have done and published also with high appreciation, one was about low dose immunotherapy in platinum resistant head and neck cancer, our institutional experience at HCG Manavata Cancer Center. And the challenge was that the standard immunotherapy doses of nivolumab, that is close to 400 milligram, is actually cost prohibitive for majority of our patients. 9 out of 10 patients could not afford it. But when we combined a low dose nivolumab along with oral metronomic chemotherapy, we realized that the adaptation had improved, the toxicity went down, and the clinical outcomes were actually equivalent as compared to those patients who are receiving the full dose of nivolumab. And this particular study we published at ASCO also in 2024. And the lesson learned was innovation in India must include cost adaptation also, and it should be biologically driven protocols rather than just individualized treatments. And the standard of care has to be taken into account, and affordability, because until unless science is used for the masses, it is not meant to be for a limited few, but its use has to be for the masses. Only then we'll be able to make a change in the lives of the patients. Next slide, please. We know that AI is actually making a fast inroads, and we were part of again an investigated trial study where it was a UK based group, where we did a study on AI based building up an AI based tool for early detection of oral cancers. It was a very simple study where each of the subjects with oral symptoms, not necessarily cancer, we took 9 photographs using simple smartphones for all these 5,000 subjects. So it was actually 45,000 photographs, and these 45,000 photographs were run through an AI algorithm. And based on these 45,000 photographs, I was astonished when the sponsors told me they developed close to 30 million algorithms. And based on these 30 million algorithms, the sensitivity of picking up early oral cancers was as high as 92%, though the specificity was around 80-81%, but that was a phase 1. It was just a beginning of the journey, and I'm sure now we are looking for a larger cohort of subjects, and we are looking for a sensitivity of anywhere 95 to 98% with a higher specificity. So probably the intention is to develop an app where anyone using a smartphone can download that app, take a selfie of your oral cavity, and the app will actually guide whether this is a pre-malignant or a malignant condition, and what is the diagnosis, likely diagnosis, and what is the best way to treat or investigate further. Next slide, please. Similarly, we were part of another study, which was OncoVerix F. It was basically innovation, and it is a very, very proud moment for us when it was published. And now they are trying to make it as a over-the-counter available test, where it was a simple test where using simple peripheral blood serum sample, various molecular signals were being identified and to detect early malignancy based on certain multi-cancer detection, high sensitivity assays using AI. It was not only circulating tumor cells, and the key findings was it was high sensitivity, close to 99%, hardly any invasiveness because just a serum sample, and the clinical utility is profound. So they have now made it available for 16 different gynecological cancers. 1 cc serum is enough to run through that assay, and that's called as OncoVerix F. F is for the female subjects as of now to begin with, and we will be able to identify the high risk subject. If it comes positive, then accordingly the subjects or the ladies should go forward with further imaging and diagnosis. And if we have a 99% sensitivity, it would be a dream come true that any one of us can just walk into a lab or into a pharmacy and say, please give me a OncoVerix F, um, and I put my blood on that, and it will guide me, something like a blood test, whether I'm having malignancy or no, something like antigen antibody reaction. And I think the world is definitely moving ahead in that direction. Next slide, please. So, as an investigator, I feel that our duty uh, is to make sure it is not just to treat disease, but to generate evidence as well. In future, every protocol has to be followed, every data point submitted, and it has to be submitted in such a way that shapes the next standard of care. And the final evolution, when genomics guides decisions and technology reduces the variability, we move from good surgery to responsible science, because it's not only about uh, operating, but it ensuring that the patient has a good survival, and we have to make oncology a chronic disease, and we have to increase the number of cures which we are able to achieve. Next slide, please. Thank you very much for this opportunity and this patient listening. Once again, InoRx for making me part of this wonderful webinar. If there are any questions, I'll be happy to take them.
Thank you so much, Dr. Raj, for that fantastic presentation where you really led all of us through your journey of how you began clinical trials at your hospital and how you embraced different advances and innovations, including using AI, using robotics, and generating a lot of evidence. I especially like the way you concluded by stating that it's not just about treating diseases, but also finally generating evidence. As doctors, each of us, if we remember this, it'll be so great that we are able to contribute together towards drug development so that we actually end up improving and saving lives. And I just thought I'll set the ball rolling by asking two questions to you, Dr. Raj. First thing is, what would be your advice to young oncologists who are beginning their career? As why should they do clinical trials at all? Obviously, most of them are engaged with the different competing responsibilities, and it definitely takes a significant amount of time apart from their clinical load. So, why at all should they do apart from this? What you said, is there any advice you would like to give specifically?
See, I think I started doing clinical trials for my own self improvisation. See, because once you come out of your teaching institute, wherever you are completing your MCH or DM, once you come out of your clinical training institute, and you are out into the open, either as a freelancing surgeon or a practitioner, you are taking decision all by yourself. many a times they are not guided. And over a period of time, we know that science is so fast evolving. What was right yesterday is obsolete today. So I started clinical trials to remain updated with science. That was my most important part. Second part is we are able to offer to our patients treatment options which are better as compared to what is already going on. So we are more futuristic in a vision. And third most important part, I would say, clinical trials have taught me discipline. Because before doing clinical trials, I was very poor when it came to documentation. It was just like operating, scribble few notes and leave it alone. But after clinical trials, see, as I began with, I have an experience of 300,000 plus cancer patients whom we have treated. When I say this, I have a data of 300,000 patients which are there with us. Right from the day they stepped in, what was their diagnosis, what was the receptor status, what treatment was offered to them, how did they progress, if they failed, why they failed, when they failed, and what was the change in therapy. So it has helped me become a better clinician. And while working as a team, protocols are set in, guidelines are being set in. So that is the reason why every young oncologist should participate in clinical trials.
Thank you so much, Dr. Raj. So well put. I also want to ask another question since you're a robotic surgeon. We all know that most of the time when it comes to these advanced devices, many of these devices are imported from the West and we are utilizing it for providing patient care. Have you noticed that new companies coming up within our country to develop these kind of more sophisticated devices to deliver care to patients in oncology?
See, as such, because if you look at the SSI mantra has been designed by a cardiothoracic surgeon. In fact, most of our new equipments, it's like we are designing either indigenously. I am now happy to be part of another robotic. It's a Chinese startup which is now it's a exchange transfer of technology, and it will be manufactured in India. So the basic viewpoint becomes that we are able to offer the same robotic technology to more number of patients rather than being it a luxury. It actually has to be a commodity. Until unless you get it out of the hands of a few surgeons or few patients or few hospitals who can afford it, we will not be able to generate evidence, we will not be able to generate numbers also. So I take pride in saying that when we started doing our robotic platform journey with CMR, when I started, I have trained more than 65 international surgeons who have traveled to Nasik to witness how surgeries should be done, how they are done, what are the protocols, and we have guided them. I have traveled abroad. I started the robotic journey of King Hussein Cancer Center at Jordan. So forget about the overseas part. Recently, I happened to operate in Goa also, robotic prostate cancer surgeries in Goa. For the first time in the history of Goa, robotic prostatectomies were done. So I think there is enough space within our country that every patient should get the best technology, best quality care.
Thank you so much, Dr. Raj, for looking at the way you shared the information here in your slides. I can easily say that you are an inspiration, certainly for surgeons in India and for clinical trialists. Personally, I've faced a lot. I've been worked with a lot of clinical trials, worked with a lot of investigators also. So it's very rare to find a clinician who is so busy like you to give so much time for research and to do it so well and to have a lot of real world evidence generation also, as you have beautifully done over the last 2 decades. So thank you so much, Dr. Raj, for giving your valuable time this evening with us and with all the delegates. There's just one question. What are the investigators' responsibilities regarding informed consent and what challenges you specifically face while doing oncology clinical trials, the consent process? So, any personal experience you have?
See, I have a very, very clear understanding, and that is how I put forward my thoughts to my clinical research team also. First, you should read the protocol yourself. And if you are convinced with the protocol, if you are convinced that if it is your near and dear one who would benefit from the trial, only then you should offer it to your patient. If you can offer the same trial to your family member or your near and dear one, that means it's a good trial for the patient. And as an investigator, while taking a consent, it is extremely important that we have to be 100% honest. We are in a country where we are very lucky that our patients trust us. Today, also, 10 patients, if I advise to be part of clinical trials, 9 out of 10 will say, Sir, tell us where to sign. If you are thinking for us, it is definitely going to be right. Whenever a patient trusts you so much, the onus of responsibility increases fourfold. So that is, as far as the patient part is concerned, taking the right informed consent. It should be a genuinely informed consent. A second part, the clinical trial ecosystem. The principal investigator, patients, they are part of the ecosystem. Equally, two other major arms. One is the sponsor, and second one is the CRO or the SMO, which is conducting the trial. So you have to come out with the best quality trial. So it is better to identify 4 subjects, but screen only one subject who is going to be sure or who is committed enough to complete the entire journey of the clinical trials. Because if you have more number of dropouts, you are actually doing more harm to science than good.
Absolutely, so well put. It's so important that we identify the right patient who's really able to complete the entire duration of follow-ups so that there are no dropouts and poor data quality. Thank you so much, Dr. Raj Nagarkar, once again. We will be moving on to the next session now. Before that, we'll have a poll here for the audience. What is the biggest operational challenge at your site when conducting oncology clinical trials? For those of you who have been doing clinical trials, feel free to choose from any of the options: patient recruitment and retention, high protocol complexity and visit burden, high protocol complexity and visit burden, then data entry, queries and documentation workload, IP handling, retaining patients for long term. Yes, please fill your responses. Yes, I think you can end the poll, Aliza, and show the results also.
More are coming through to us. 1 second, Dr. Melvin, 1 second.
Okay. Okay.
A lot more.
I think in India,
Yes, audience.
in
Yes.
India, the patient recruitment is not a challenge. The Moja part is
Yeah, I agree.
not a challenge.
Yeah,
It's
I
about
agree.
how
Yeah,
complex it is.
especially in oncology. Yeah.
So I liked it that a lot of people have very honestly said protocol complexity is a challenge, particularly when it comes to oncology clinical trials. It's getting more and more sophisticated. Good. Yes. So I think we'll end this poll here. So from this poll, there's just one thing I wanted to say, that there are obviously a lot of issues that an investigator faces when doing clinical trials, a lot of challenges that they encounter while doing clinical trials. So being an investigator for a multicentric global clinical trial is obviously not a joke. Definitely, there are lots of responsibilities. One has to be very judicious in the way they end up performing a clinical trial, because Obviously, on one side, we're trying to deliver novel technologies to patients. At the same time, with putting ethics as the highest prerogative, and at the same time ensuring that you're generating quality data so that tomorrow when an FDA or EMA or a CDSCO inspector visits our hospital, we'll be able to really substantiate or validate the data that oh, it is really a good quality data, and so they can rely on that data. So we have to discuss all these issues on responsibilities of investigator, especially from the latest guidelines of ICH GCP E6R3. We have with us Dr. Vinayaka, whom I earlier introduced. So over to you, Dr. Vinayaka, to take over the next session.
Thank you, Dr. Melvin, for a quick introduction. So I would like to welcome all once again for this webinar. So my topic for today's discussion, or the presentation rather, is the investigator's responsibility, especially with reference to ICH GCP E6R3. Obviously, we are well aware of the attendees who are attending this session are pretty much aware as far as investigative responsibilities are concerned. So my focus of today's presentation will be basically to take you through what are the key changes which have been noted, I mean to say, incorporated in the R3 guidelines as we compare to the R2 version of the ICD-GCP. So during this session, yes, I'll be focusing what are the key changes, basically, which ICBCP R3 is talking about, the quick definitions and roles and responsibility, the quality by design strategy and the risk management, how we can implement in the conduct of the clinical trials, what are the key responsibilities as far as investigators, again, mainly focusing on the R3 guidelines, and what are the common inspection findings and how QBD can help us in managing or preventing or taking care of those observations in the future. Yeah. So, yes. GCP principles basically are intended to support efficient approaches to trial design and conduct. One of the key aspect when you talk about IC GCP E6R3 is basically it fosters quality culture. It fosters quality culture. It is also basically it focuses more on quality by design strategy, not only at the planning stage but also during the course of the clinical trial. So the objective basically is to proactively identify the critical to data critical to quality factors. And once we identify those critical to quality factors, obviously we can identify those key risk factors which can impact those CTQs and have a risk mitigation strategy in place. So the approach as far as ICH GCP E6R3 is concerned is risk proportionate. Trial conduct is essential. That is, the investigator should oversight and the risk mitigation measures should be proportionate to the importance of data and the level of risk to the patient. Basically, what it says is the extent of the oversight by the investigator should be proportionate to the risk involved in the clinical trial. And in case if there are any complicated processes, for example, IP administration, obviously such procedures will require key closer oversee by the investigator as compared to the other aspects. So the ICL GCP E6R3 also focuses on engagement, early engagement of the participants, especially the key stakeholders, that is patients, communities, advocacy groups, healthcare professionals. Now, why it is important is at the planning stage itself, if we can anticipate the issues which the patients communities, the patient advocacy groups and the healthcare physician, obviously we'll be able to address those. We will be able to upfront anticipate those risks and have a mitigation plan very well built into the protocol itself rather than later on addressing it as we conduct the clinical trial. As one of the aspects which Dr. Raj spoke about, that yes, it's important to as far as consent is concerned, it's important to clearly inform them what are the key aspects. And that is where one of the key challenges, one of the question which came up was, what are the key challenges in conducting oncology study and the complexity of the clinical trials That is something which can be addressed right at the beginning of the clinical trial when we are designing the study. When we take key inputs from the participants, the patient advocacy groups, they are the ones obviously which can give key inputs as far as the complexity of the protocols are concerned. If there are too much of visit burden, obviously that can have an impact on the patient compliance as well as patient retention. So these are things which can be taken up upfront when we are designing the clinical study by engaging with these stakeholders. And finally, yes, the protocol systems processes, whichever will be implemented as part of the clinical trial, those should be fit for purpose. So as per the requirement, those investigations and the assessments should be planned in the protocol. Right now, the current challenge is most of the protocols will try to have answers to a lot of questions, research questions in the same study, as a result of which a lot of investigations, a lot of assessments are planned in the same protocol, which tends to burden the patients, as a result of which, yes, so what we see is a lot of patients dropping out of the study, a lot of patients losing the motivation to continue the participation in the study. So what is important is the protocol should address the key objectives accordingly. The procedures and the investigation should be planned in order to ensure that at the end of the study, the desired outcome, the desired data is available based on which the further clinical trials can be planned. Uh, Now, coming to the investigators, I'm interested in the definition. I'm sure most of the people are aware. Investigator basically means a person responsible for the conduct of the clinical trial, including trial participants for whom person the investigator is responsible for the conduct of the clinical trial. Obviously, the investigator should be qualified by means of education, training, and experience, and should be familiar with the use of the investigational product by means of having good knowledge as far as in investigator brochure and any other product related information which is provided. What does ICH E6R3 in particular with respect to the investigator responsibility talks about? This is covered in section 2, right from section 2.1 to 2.13. So these are on the left, these are the key roles the investigators plays as far as the conduct of clinical trial is concerned. And yes, so most of them, obviously, I'm sure most of you people would be aware of. My today's session would focus mainly on those challenges or those key changes which R3 is speaking about. So that is something which I'll talk more into. So QBD is something which we have been discussing, but what is quality by design and how it can be incorporated in the clinical trial protocol by thereby ensuring we minimize the risk, at the same time improving the data quality as well as ensuring patient protection. So quality by design strategy is basically nothing but identifying the critical to quality factors basically at the initiation of the clinical trial, means right from the planning stage of the clinical trial, when we are planning the study, when we are strategizing the clinical trial, that is when we will engage all the stakeholders and try to identify the critical to quality factors which can have an impact on the overall study outcome. So once you identify those critical to quality factors, obviously you need to identify what are the key risks which are the key risks which can impact those critical to quality factors. Once you have identified those key risk factors, you need to have a mitigation strategy in place. I'll discuss more on that in the upcoming slide. So basically, we need to have strategies which should be implemented as part of the quality by design to avoid, detect, address, and prevent the recurrence of serious non-compliances. At the same time, ensuring that we are collecting the key data which is required as far as the outcome assessment is concerned. So the QBD should be implemented to identify the factors that are critical to quality, thereby ensuring that the study is conducted with the highest quality, thereby and also minimizing the risk to the trial participant and also to the data quality. So this is the QBD framework. That is where at the draft stage, when we are designing the clinical trial, that is where we need to engage, okay, as part of step one, all the key stakeholders. Okay, as we discussed earlier, it's not only the investigator or the sponsors, the experts. Also, it's important to engage the patients, the patient advocacy groups, community, healthcare professionals in taking the inputs as far as understanding the critical to quality parameters are concerned. Once we identify these critical to quality parameters, it's important. It will be easy for us to understand what are the key risk factors which can impact those critical to quality factors. And once we have those, we can have the mitigation plan in place. Just to give you an example, especially for oncology studies, okay, we all know that overall response rate is one of the primary endpoints, one of the key endpoints for the study as part of efficacy assessment. Now, the critical to quality factors as far as the objective response rate is concerned, that is the efficacy assessment. Efficacy assessment is concerned is basically the radiological assessment. So if this radiological assessment, if it is not properly conducted, obviously it can have a direct impact on the assessment of the overall response rate. So what are the risks which can have an impact on the radiological assessment? Those are nothing but those are the risk factors which we need to identify, such as variability in the radiological assessment. Not to mitigate that, obviously we need to have a protocol, that is our radiology manual in place, wherein which, for example, the proper guidance and the guidelines are given. For example, for oncology studies for tumor assessment, we need a tumor assessment. We need to standardize the radiological assessment as per REGIST 1.1 guide, REGIST 1.1. So as per REGIST 1.1, obviously all the site needs to uniformly follow. In addition to that, in order to avoid site-to-site variability, we will be also engaging a central radiologist, so that central radiological assessment of the tumor assessment, thereby reducing the site-to-site variability. So this is how you mitigate the risk. Okay, so at one side, obviously we have identified as overall response rate as an important endpoint for which the critical to quality factor, obviously in this case, would be tumor assessment. And for tumor assessment, obviously one of the critical to for tumor assessment, which is the critical to quality factor, the risk is basically the inappropriate radiological assessment. And how we'll mitigate it? By ensuring that we uniformly conduct those radiological assessment, and thereby and also deploying a central radiology, thereby ensuring that we minimize the variability, thereby protecting the primary endpoint, that is the overall response rate. And that is how we mitigate those risks upfront when we are designing the clinical trial. So this is how the quality by design strategy can help us to prevent the I mean to say the deficiency or the variability in the clinical study. So this is just one example. So quality by design strategy can be deployed at the start of the study as when we are designing the clinical study. As we continue to conduct the clinical study, even at that stage, during the conduct of the study, also QBD strategy can be deployed. For example, yes, so we all know that once the study is started, one of the key aspect is which can impact the primary endpoint, that is efficacy endpoint, is the patient dosing. Now it's important that we ensure that there are no discrepancies or errors in the patient dosing. So that is something which is a critical to quality factor, that is the IMP dosing. Now, how we'll ensure that the IMP dosing is appropriately managed? In that case, what are the risks which are involved in the proper IMP dosing? Those are the risk factors, such as the wrong dosing being done, or inappropriate infusion to the patient, or inappropriate infusion preparation. So these are the risks, and we can have a mitigation strategy where we can have a proper pharmacy manual which will clearly indicate how the dose calculation should be done, how the infusion should be prepared, and how the infusion should be given to the patient. At the same time, having a quality control procedure, thereby ensuring that this risk can be minimized. So this is how basically we will be anticipating the risk upfront as part of quality by design strategy, and will be having a mitigation strategy. And the step 3 is basically to ensure that whatever learning we had at the end of the study, those should be deployed in the subsequent studies which we'll be conducting, either in the similar kind of therapy areas or in the other therapy areas. But as per there would be the common risk which will be noted, those can be deployed. And similar kind of studies like oncology studies, whatever risk or whatever knowledge which we had based on these quality by design strategies, those can be deployed. Like critical to quality factors which have been identified, those can be extrapolated into the other studies as well. So quality by design strategy can be deployed even at the site level, and this is how, as part of the execution, the site can practically implement these strategies, so that we can upfront anticipate what are the risks associated by conducting the study, and thereby ensuring those risks can be upfront mitigated as part of this design. Now coming to the other aspects, the other responsibility as far as investigator is concerned. So my focus would be especially on the R3, the changes with respect to the R3 version. So as far as study delegation and oversight is concerned, Aarti talks about the investigator roles and responsibility. Investigators should clearly define the roles and responsibility of each of the study team members in the delegation law. But in addition to that, it's important that the investigator, in case if the investigator is outsourcing any clinical trial activity, be it vendors, for example, labs or the archival agencies and so on, it's important that even those roles and responsibility and the delegation should be clearly documented in the agreement. And not only that, the investigator should have a clear mechanism for overseeing the conduct of the clinical trial, not only at the site, at the study site, but also at the vendor site where the investigator has outsourced the clinical trial activity. So individuals who will be conducting the clinical trial as part of the investigative team, obviously they should be adequately qualified, trained, and adequately informed about the protocol, IP, and the assigned responsibility. But the final responsibility of the study conduct ultimately remains with the investigator. So it's not only with respect to the conduct of the study at the site, but also if the investigator has outsourced those activity to any other third party, any other external service provider, such as lab, archival agencies, and so on, the investigator will be ultimately responsible for the data or the activities performed by those external agencies as well. So this is a new update as far as R3 is concerned with respect to the engaging the external service provider, at the same time oversee the conduct of those service providers as part of the R3. Yeah. So next is the informed consent. Yes, yeah, the previous R2 version did talk about informed consent. It's not something new, but what R3 talks about is the technology which can be incorporated as far as informed consent is concerned. That is electronic informed consent, e-informed consent. So as informed consent is becoming more and more intensive with respect to the communication, with respect to the information which being shared to the patient, it is important that all the aspects of clinical trials are adequately informed to the patient during this process, since once the patient is fully aware of the study requirements and what is expected of the patient, obviously there's better compliance from the patient, and it also reduces the chances of dropout. Now, yes, as part of electronic informed consent, the modern consent do have the visuals, videos, interactive tools to improve patient comprehensions, which is important as we discussed, not to improve the patient retention. Now, it's important that all these aspects, the visuals, videos, interactive tools, should be submitted. First of all, we should have the ethics committee approval for all these text, videos, interactive tools, and all. In addition to that, as far as the e-consent is concerned, the procedure of administering the electronic consent should be completely reviewed and approved by the ethics committee. Only then we will be able to adopt the e-consent. In addition to that, uh, the electronic informed consent, in case if it is adopted for a particular clinical trial, it's important that uh, uh, the patient should be comfortable. The patient should be comfortable in utilizing this e-consent, should be comfortable in utilizing the e-technology as part of the informed consent. If they are not comfortable, there should be alternative systems available. I mean to say, obviously, paper informed consent as an option should be available for them so that they can provide their informed consent. It should not happen that just because of technology, they cannot participate. Such instances should not take place. That's what R3 talks about. The participant must be offered alternative method. If they are not comfortable with the digital tools, the paper-based consent can also be obtained. And most importantly, as part of R3 is with respect to the informed consent is the patient identity. Yes, there should be proper process in place regarding how to verify the patient identity as part of the informed consent documentation, and that is something which the investigator has to take care of with respect to the consent. Coming to the data governance, now, yes, this is something new which was not part of the previous R2 version. So data governance is basically both the investigators and the sponsors are responsible to ensure they deploy processes which shall take care of the data integrity, the data traceability, as well as data security. Okay, the data governance part specifically states that the investigator should have processes in place, to ensure the protection of confidentiality of patient trial data, ensure that processes for managing computerized system to ensure that they are fit for purpose and are used appropriately. So in case of computerized system which are being used, for example, e-consent, be it e-qual, e-pro, e-pro, and so on, or any each EHR, let's say, for example. So in that case, even if those tools are used, those should be fit for purpose. They should be able to capture the data for which those tools are developed, and no other irrelevant information should be captured. That is what in ICH E6R3 talks about. Also, the investigators as well as the sponsors should have processes to safeguard essential element of clinical trials such as randomization, dose adjustment, and blinding. As we discussed earlier, obviously all these processes can be anticipated upfront as part of the quality by design approach, wherein which the risk associated with these procedures like randomization, dose adjustment, blinding can be upfront anticipated, and a process that is a risk mitigation process can be in place. Um, um, as part of the SOPs, the investigator should have a process to support key decision making, such as data finalization prior to analysis, that is, approving the case report form, ensuring prior to database lock, the investigator should be able to approve all the case report forms prior to analysis. There should be processes for unblinding as well. In case of any emergency unblinding, there should be proper processes in place. Allocation of analysis data set. Yes, as part of at the end of the study, as per after the database lock, there should be adequate processes which clearly define as part of the statistical analysis plan which patient would be part of the safety data sets, efficacy data set, or any other data, for example, PK data sets, and so on. So yes, the processes should take into consideration the changes in the clinical trial design, and that is what how the data governance framework should be built into. As far as data lifecycle elements is concerned, the
By
data
closing
governance
by
also talks about.
another 5 minutes.
Sorry, sorry.
Okay, Vinayak,
Yeah.
you can
So
try
yeah,
completing another
yeah,
5 minutes.
yeah. So as part of data governance, I see DCP E6R3 also focuses on data capture. Not only about data capture, it also ensures that all the relevant data, metadata should be properly available. In addition to that, there should be proper review process for this metadata. In case if there are any data corrections, obviously there should be proper metadata to ensure that those any changes or corrections are attributed to the person who has made it, along with date and timestamp. The system should allow data transfer, exchange, and migration without impacting the data integrity or security or traceability. Also, there should be adequate processes as part of the data governance, wherein the finalization of data set should be done prior to the analysis. As we discussed earlier, the process should be in place where the patient I mean to say the safety set, the efficacy set, those analysis sets should be upfront finalized before we go ahead with the analysis. The investigator should have adequate procedure in place for the retention. Okay, because as part of the regulatory requirement, records are to be retained for a long term. Okay, and those should be also accessible during the archival period. So certain regulatory agencies may require 5 years, some may require 15 years, some may require 25 years, 25 years. I mean to say. So during that period, the investigator should ensure that the documents are retained till that period. Not only that, those should be accessible. In addition to that, especially with respect to this electronic data, these documents should also be endurable during this period. And the investigator should ensure that these requirements are met. And before destruction, obviously, including the electronic data, the sponsor's prior approval should be taken. Only after that, the investigation can do the destruction. Coming to the IMP management, yes, this is pretty standard with respect to accountability, handling, and administration. There should be proper documentation. But sponsor can help as far as IMP management is concerned to the investigator by setting up the electronic system such as IWRS, which will be able to allocate the drug to the investigator based on which the investigator will be able to dispense this medication to the patient. Yes, one more important aspect as far as the IP management is concerned is investigators should closely oversee the IP management processes at the site. As we discussed earlier, IP management is something which is critical that can have a significant impact on the study outcome in case of wrong dosing or if the drugs are not stored. So these are the procedures which should be closely overseen by the investigator, and yes, close oversight should be done. Another important aspect as far as the IP management is as far as the R3 is concerned is the guideline talks about shipping the product to the patient location or location closer to the patient. Not only that, the site team can provide or can administer the drug at the patient location, or either the patient themselves or the healthcare physician can also administer the drug. But these were things which were not covered as far as the R2 version was concerned. Finally, maybe after the end of the study, it's important that in case if the patient is completing or in case if the patient is dropping out of the clinical trial, whatever data is collected, that data should be utilized as part of the analysis. It should not happen that if the patient has not completed the study, that data should be discarded. Also, it's important to preserve the data integrity. So as we discussed, once we have that data, even if the patient have dropped out, uh, whatever data which has been collected, that should be analyzed, because in case if it is not analyzed, that can bring in biasness and can impact the reliability of the trial conclusion. So the guideline insists that even if the patients have dropped out, have not completed, that data should be included as part of the safety analysis. In addition to that, even that data can also be used for the efficacy assessment. In case if the patient has completed at least one post-baseline efficacy assessment visit, those data can also be included as part of the efficacy outcome assessment. Another important thing which this guideline talks about is the investigator should make reasonable effort to understand why the patient is withdrawn, and should try to explain to the patient the value of patient's continuation in the clinical study, because obviously, in case if the patient drops out, and similar such situation happens among other patients also, obviously we'll not be able to evaluate the study outcome, and that is what something which has to be explained, and how the study can be beneficial to the patient and the society at large. Having said that, it should not happen that the investigator team is unduly influencing the patient decision. But yes, efforts can be taken to try to explain the patient the importance of patient's continuation in the clinical study. Results in publications, yes. The guidelines talks about the study should be registered on the public registries such as CTRI as far as India is concerned. As far as US, we have clinicaltrials.gov, and for Europe, it is Udacity. So these guidelines recommends rather that the study should be registered. Not only the registration part of the study is concerned, but also results. The results should be disclosed on these registries in a timely manner, thereby ensuring transparency and gaining the public trust in clinical trials. In addition, at the end of the study, the investigator should consider sharing the trial results with the patient, obviously, in unbiased and non-promotional way. The study results, along with the treatment arm in which the patient was, that can be disclosed to the patient. So some of the common USFDA inspections as far as at the site, which are noted by the in the recently released USFDA report, was inadequate investigator oversight, okay, of the delegated study activities, failure to follow approved protocol, deficiency in the informed consent, okay, failure to protect patient participant safety and welfare, poor documentation and record keeping process, especially with respect to medical history, which is very common finding, improper control of investigational product, and failure to comply with the regulatory requirements such as delay in safety reporting, that is SAE reporting and so on. So all this, deficiencies, especially most of these deficiencies can be upfront addressed if we deploy the quality by design approach at the start of the study and also during the conduct of the study. Similarly, EMA has recently released the report during which yes, these are the common findings, and based on which the recommendation of EMA has been clear rules and agreements are essential at the investigator side. As we discussed, investigator should have proper oversight, and those oversight should be obviously proportional to the rest of the procedure. And the oversight should not only be at the site but also at the service provider end as well. Also, the access to the source data and medical record should be available. And again, this is a major issue, especially with respect to medical history. As I told you, at times the medical histories are not available, as a result of which the patient's eligibility cannot be confirmed. The regulatory expectation is basically to have these medical records right from the time of diagnosis of the disease itself. And after that, whatever treatment the patient has received, all that information should be part of the medical history. Also, the recommendation is to ensure proper management of essential trial documents, which is obviously it's important to ensure that data credibility as well as data integrity can be assessed. Strengthen the trial management and protocol compliance, which is pretty standard. ensure adequate training and supervision of the site staff, and improve data integrity and documentation practice. So basically, EMA inspection repeatedly shows that most deficiencies occur not because of the investigator's lack of knowledge, it's because of the poor oversight and documentation practices. And that is what we can address as part of the QBD approach. We can definitely address these strategies. So in conclusion, yes, investigator ultimately remains accountable for the patient's safety, data integrity, and trial conduct, regardless of the delegation or technology used. And it's important, as we discussed, it is important. It's not only about the trial team, but also the service provider oversight should also be done. Active oversight is the cornerstone of the investigator responsibility. QBD and risk proportionate oversight are central to modern trial conduct, and patient centricity and the processes should be fit for purpose in order to ensure that the study is feasible and we obtain quality data and meaningful trial outcomes. Now, InoRx, as far as the QBD capabilities are concerned, we have a group of enriched and experienced professionals who have experience in diverse therapy areas, especially in the area of oncology. And we have the mix of both medical as well as quality experts, having more than 2 decades of clinical trial experience. We do actively engage with HCPs and also have access to the patient advocacy group. And obviously, we have significant technology to support us as far as digital technology, as the data acquisition is concerned. And also, we have critical to quality libraries with us, which can help us to design a better clinical trials using the quality by design approach. So with this, I would like to end my presentation. Thank you. And if there are any questions, I would like to take those questions.
Yeah, I think you could put, Aliza, you could put the poll now. And meanwhile, Dr. Vinayak, I just have a few questions. Maybe I'll start with just one question first to you and then take the questions from the audience. You have been in the industry for so many years, and I'm sure you have been interacting with a lot of investigators from different regions in India. So, do you have any good anecdotal experiences to say about your experience with any particular investigator, principal investigator, any good practice that they followed, which something can be said as a role model for others to emulate?
Yes, over the last 2 decades of the clinical trial experience which I have, yes, I have come across multiple investigators, in fact, multiple sites. One of the key differentiators of these sites being, obviously they have robust processes. Okay, even though at the times where SOPs were not required or mandated for conducting the clinical trials, they had set processes. They set processes, and as a result of which, they used to, as we discussed today, they had set processes for the informed consent, for ensuring proper documentation, and I mean to say, ensuring patient retention as well. Now, documentation as far as SOP is important. In addition to that, the having robust site team, okay, having good team, that is also important, because if the site is having a good team, that is when whatever issues which we have, maybe we have adequate backup, they'll be able to address those issues upfront. And so the key process is what I understand from all my experiences, having good SOPs, sites having good SOPs, having good team structure, having good oversight, basically the investigators actually, which was stand apart as the good process is what was followed was the investigator himself or herself was keenly interested in overseeing the conduct of the clinical trial. Not only that, engaging with the patients upfront, not only just at the time of enrollment, but also during the follow-up with it. As Dr. Nagarkar said, it's important. It's important to ensure that, okay, the team which is engaged, they should be convinced whether their relatives will be able to participate. So it's important that the team is adequately trained. They should be thorough with the protocol. Only once they are trained and they know what are the risks and the benefits associated with the study, they will be able to properly carry out the procedure. They will be able to properly explain to the patient. If the patients are adequately motivated, obviously the dropouts can be less, and yes, they will be happy to continue the association in the study. Plus, the protocol deviation. One of the key aspects at the site of those centers who have been good in conducting clinical trials is the protocol. protocol compliance. So what these sites used to do is they used to have periodic review meetings of protocol deviations. Also, not only protocol deviation, also adverse events review meetings they used to have. So that what happens is they also kind of did the trending analysis. Once they understand where they are going wrong, also they had a proper kappa in place, so that these are the good practices they have deployed. Okay. In addition to that, even the adverse event monitoring, periodic review of the adverse event help them to understand what are the adverse event basically they are noticing as part of the particular clinical trial, and whether there can be ways and means to prevent those by pre-medication or by prophylactic treatment, I mean to say, or by any other measures. For example, upfront counseling the patient. Upfront counseling the patient that yes, if so and so adverse events are expected, they should report to the center so that upfront immediate treatment can be initiated. So these are ways and means. Some of the good centers are following the practice. Yeah. So these are things. So it's not only about patient recruitment, but also having proper robust processes in place so that we address those issues upfront, or we upfront anticipate those issues and have those processes in place.
And just one more question. From a sponsor's perspective, do you think that a CRO or sponsor should implement any specific measures at the site to help the clinicians, especially the PIs, to implement the R6, E6R3 guidelines at the site?
Yes. So as we discussed earlier, okay, we have the team. Okay, we have the team which can help, because what is important is, as far as this QBD approach is, try to have we should have a library of critical to quality factors upfront before starting the study. We can engage with the site. We can help them to identify what are the critical to quality factors. So maybe even if the site is not being engaged at the start of the clinical study, even during the conduct of the study, just prior to start of the clinical study, that is at the time of site initiation, we as a sponsor or CRO, we can sit with the site, try to help them to identify what are the critical to quality factors for those projects. Like I told you, overall response rate is one of the important endpoints. Then in that case, one of the critical to quality factors is the tumor response assessment, and radiological assessment happens to be an important parameter, the assessment, which can have an impact on this tumor assessment. So in order to minimize that variability, we can standardize it. We can help them. We can have a central radiology. So like that, at the site, maybe the dosing, there will be significant errors as far as the IP dosing is concerned. So we can help them in similar way. What are the critical to quality factors associated with that project? And once we identify that, then yes, risk, we can upfront identify a proper, do a proper risk assessment and have a mitigation plan in place. So yes, we can closely work by following these processes. Yeah.
Yeah, thank you, Dr. Vinay. So there are also some comments about multivariate analytics. So obviously, multivariate analytics is extensively used in oncology trials, especially to find what are the factors which influence the response to therapy, and also advanced clinical trial designs, including factorial designs. They're all used extensively to make trials more efficient. But for two reasons. One is it's being outside the scope of this particular webinar, and the fact that we're already late, we will be closing this session and not taking further questions. But I would like to once again thank Dr. Vinayak for guiding us through this session about ICH DCP 6R3 guidelines in the context of responsibilities of a principal investigator. So thank you, Dr. Vinayak. And now, once again, thanks to all the delegates who have been part of today's webinar. I now hand over the time to Alisa.
Thank you, Dr. Melvin, and thank you, Dr. Vinayaka. It was such a great presentation. We're very grateful that Dr. Raj was able to also join us. And thank you all for staying on this evening. We hope that you enjoyed the content. Just wanted to flash this slide up here so that you can see what's up next for us when it comes to the Academy. And our next webinar will be taking place on March 28th. So we'll be sending out information on how you can register for that, as well as some of the future ones, just so that you can see. Our next one will be very great. We're having some of our folks from Novo Nordisk join us from Switzerland, so we're very excited for that conversation and topic. and some of the other upcoming ones are going to be great as well. So if you attended today, you'll be receiving a certificate that shows you had participated in today's session. So look for that in the next few days. And if any questions come through or come to your mind, please just go ahead and email us at hello@inorx.com, or you can feel free to drop anything in the chat and we'll make sure to get back to you. But we really appreciate everybody's time and hope that you found all the content to be very helpful, and we really appreciate you for joining.